We have seen how the membrane changes shape, how the cytoskeleton generates force, and how junctions connect a cell to its neighbors and to the matrix. But none of those structures is permanent. Receptors, adapters, motors, junctional proteins, and membrane components are continually being made, modified, delivered, and eventually removed. A functioning cell, therefore, needs more than a set of parts. It needs a system that manages the life cycle of those parts. That system begins with a basic division of labor. Some proteins are made in the cytosol. Others enter the endoplasmic reticulum, travel through the Golgi apparatus, and are sent to a membrane, an organelle, or the extracellular space. Material that has reached the end of its useful life then enters a disposal route. So the question for this section is not simply where an organelle is located. It is what job that compartment performs, where its cargo comes from, and what happens when the system cannot keep up. Look at the cell as a set of specialized workspaces. The nucleus stores and reads genetic information. The cytosol supports metabolism, transport, and translation on free ribosomes. Rough endoplasmic reticulum makes proteins that will enter a membrane, an organelle, or the secretory pathway. Smooth endoplasmic reticulum changes lipids, handles hydrophobic compounds, and stores calcium. The Golgi apparatus modifies and sorts cargo. Endosomes receive material brought in from the plasma membrane. Lysosomes digest internalized material and worn-out cellular components. Proteasomes deal with selected cytosolic proteins, while peroxisomes specialize in oxidative reactions, such as the breakdown of very long-chain fatty acids. This separation is not merely architectural. It allows enzymes to work at useful concentrations without exposing every part of the cell to their activity. A lysosomal enzyme can be highly effective in an acidic compartment without digesting the cytosol. A protein can be modified in the ER and Golgi without interfering with unrelated cytosolic reactions. Once the cell is divided into these workspaces, the next problem becomes routing. How does a newly made protein know where it belongs? The endoplasmic reticulum synthesizes all transmembrane proteins and lipids for the plasma membrane and cellular organelles, and it is the initial site of synthesis for secreted proteins. The rough endoplasmic reticulum is not simply a storage surface for ribosomes. It is the entry point for a defined class of proteins. Proteins intended for the cytosol are translated on free ribosomes and remain in the cytoplasm. Proteins intended for the plasma membrane, another organelle membrane, or secretion carry a signal sequence near the beginning of the nascent chain. Translation begins on a free ribosome, but the signal sequence directs the complex to the ER membrane. The ribosome docks there, and the growing polypeptide is threaded into the ER lumen, or inserted into the ER membrane as it is translated. This creates an important topological distinction. A protein placed into the ER lumen is entering a space that is continuous, in topological terms, with the extracellular environment. A membrane protein is inserted with a defined orientation that can be preserved as it travels through the secretory pathway. In other words, the ER does not just make the protein, It establishes where the protein will face and which route it can take next. The destination is being encoded while the protein is still being born.
Once a new protein enters the ER, it is not immediately sent onward. It has to acquire the conformation that allows it to work. Proteins can fold into their active shape, form disulfide bonds, and assemble with other polypeptide chains into larger complexes. Sugar groups, called N-linked oligosaccharides, can also be attached to specific asparagine residues. Chaperone proteins help this process. They hold newly made proteins, prevent inappropriate interactions, and retain them in the ER while folding and modification are still incomplete. That retention is important because a protein that leaves too early may reach the plasma membrane or extracellular space in a non-functional form. The ER is therefore doing two jobs at once. It is the manufacturing site for a major class of cellular proteins, and it is the first inspection point for those products. A correctly folded protein can enter transport vesicles and move toward the Golgi. A protein that has not reached the correct conformation remains under supervision. This is why the ER is closely connected to cellular homeostasis. The cell is not only producing proteins, it is deciding which newly produced proteins are fit to continue. A useful example is the common CFTR mutation associated with cystic fibrosis. In the mutant protein, deletion of the phenylalanine at position 508 changes the folding behavior of the newly made polypeptide. The protein does not achieve the appropriate conformation. Because the ER quality control system recognizes that problem, the mutant protein is retained rather than being efficiently sent to the cell surface. It is eventually degraded. The result is reduced surface expression of CFTR, even though the cell may have produced the corresponding messenger RNA and begun protein synthesis normally. Notice what this example teaches. A protein can fail to function at the membrane for a reason that begins much earlier, inside the ER. The defect is not simply a missing receptor sitting on the surface. It is a failure of folding, inspection, and trafficking. This also explains why the ER cannot tolerate unlimited misfolded protein. One abnormal molecule can be retained and removed. A large accumulation creates a different problem. The quality control system itself becomes stressed. That is the transition from an individual folding error to the unfolded protein response.
When misfolded proteins accumulate beyond the ER's capacity to edit and degrade them, the cell enters ER stress. The protective response is called the unfolded protein response, or UPR. Its priorities change. Overall protein synthesis is reduced, so fewer new chains enter an already overloaded compartment. At the same time, production of chaperone proteins increases, giving the ER more help with folding and quality control. The cell is trying to lower the incoming workload while expanding its ability to process what is already there. If that adjustment succeeds, the abnormal load can fall and homeostasis can return. If the overload persists, the response becomes dangerous. The failure to correct the accumulation can activate apoptosis. This is an important pattern in cell biology. A compartment that normally protects the cell can become a source of stress when its workload exceeds its capacity. The ER is therefore not just a tube where proteins pass through, it is a regulated decision point. Continue synthesis, slow the system, increase chaperones, or, if recovery fails, activate an orderly cell death program.
The ER is not defined only by ribosomes. Smooth endoplasmic reticulum, or SER, lacks the ribosome-studded surface of rough ER and performs a different set of jobs. In many cells, it forms a transition zone that generates transport vesicles carrying newly synthesized proteins toward the Golgi. In specialized cells, it becomes much more prominent. Cells that make steroid hormones use smooth ER for lipid-related synthesis. Hepatocytes use it to modify hydrophobic compounds into forms that can be handled and exported. Repeated exposure to compounds metabolized by the cytochrome P450 system can even produce smooth ER hyperplasia. Smooth ER also stores calcium. Calcium can be released into the cytosol to regulate signals and cellular responses, then sequestered again. In muscle, the specialized smooth ER is called sarcoplasmic reticulum, and its cycles of calcium release and uptake help coordinate contraction and relaxation. So, the same membrane network can support protein production in one domain, lipid and drug metabolism in another, and calcium control in a specialized cell. The next question is how cargo leaving the ER is processed and assigned its final destination. From the ER, proteins and lipids enter the Golgi apparatus. The Golgi is built from stacked, flattened compartments called cisternae. The cis face lies close to the ER. The trans face points toward the plasma membrane and the rest of the cell. This orientation creates an ordered processing line. Cargo is modified as it moves from cis toward trans. N-linked sugar groups that were added in the ER can be trimmed and extended step by step. Other sugars can be attached to serine or threonine residues in a process called O-linked glycosylation. The cisternae themselves can mature and progress from one side of the stack to the other, while small vesicles help maintain the different enzyme environments in each region. Golgi-resident enzymes can travel in the opposite direction to preserve the identity of the earlier cisternae. The result is not random mixing. A newly made protein enters at one side, is chemically edited in sequence, and emerges from the trans side with a more specific molecular identity. That identity will help determine whether the cargo goes to a lysosome, a plasma membrane, another organelle, or a secretory vesicle.
At the trans side of the Golgi, processing becomes routing. The trans-Golgi network sorts proteins and lipids and dispatches them to other organelles, the plasma membrane, and secretory vesicles. Lysosomal enzymes illustrate how precise this sorting can be. These enzymes are made in the ER and then receive a mannose 6-phosphate tag in the Golgi. A mannose 6-phosphate receptor recognizes that tag and helps package the enzyme into vesicles directed toward the lysosome. Other cargo takes a different route. A membrane protein can be sent to the plasma membrane. A soluble product can enter a secretory vesicle and be released outside the cell. A protein intended for another intracellular compartment can be delivered there instead.
The Golgi is therefore not just a finishing station. It is a sorting center that reads molecular labels and converts them into physical routes. This is especially visible in cells specialized for secretion. Goblet cells have a prominent Golgi because they export large amounts of mucus, and plasma cells show a conspicuous perinuclear Golgi because they produce and secrete antibodies. Once lysosomal enzymes have been routed correctly, we can ask what the lysosome receives and how it breaks that material down.
Lysosomes are membrane-bound compartments containing roughly 40 different acid hydrolases, proteases, nucleases, lipases, glycosidases, phosphatases, and sulfatases work best in an acidic environment, around pH 5 or lower. Material from outside the cell reaches this compartment through fluid phase or receptor-mediated endocytosis. A vesicle formed at the plasma membrane first joins an early endosome. From there, cargo can be recycled back to the surface, or it can move into a late endosome. These compartments become progressively more acidic, and the internalized material is eventually delivered to a lysosome. This route is sometimes called heterophagy because the lysosome is digesting material that came from outside the cell. The products of digestion can return to the cytosol and support nutrition, while undigested residual material can leave by exocytosis. The pathway is therefore not simply a one-way disposal chute. It includes sorting, recycling, digestion, and recovery. A receptor can be returned to the membrane for another round of uptake, while its ligand may be sent for degradation. The same endosomal system can preserve useful surface machinery and prevent an internalized signal from continuing indefinitely. But endocytosis is not the only way a lysosome receives cargo. A professional phagocyte uses a larger version of this logic. In phagocytosis, the cargo is too large to enter through an ordinary endocytic vesicle. A professional phagocyte, such as a macrophage or neutrophil, recognizes a microbe or a large fragment of debris and extends its membrane around it. The particle becomes enclosed in a phagosome. That phagosome is not yet the final digestive compartment. It must move through the intracellular pathway and fuse with a lysosome. The resulting phagolysosome brings the internalized material together with acid hydrolases and an acidic environment. Notice the continuity with the cytoskeleton video. Actin helps form the cup and close the phagosome. The membrane creates the compartment. The lysosome supplies the degradative machinery. Three systems cooperate, but they do not perform the same job. The warm phagolysosome can then kill and catabolize microorganisms. The products that can be reused are returned to the cell, while residual material follows the appropriate exit route. This is heterophagy on a large scale. Material from the outside is captured, enclosed, delivered, and digested. The next route is different because the target is not outside the cell. It is an old organelle or a damaged protein complex that the cell itself needs to clear. Autophagy is the route used when the cell needs to remove its own worn-out organelles or large denatured protein complexes. Products of autophagy-related, or ATG genes, help gather the target inside an expanding double membrane derived from the endoplasmic reticulum. As the membrane closes around the cargo, the structure becomes an autophagosome.
The autophagosome is marked by LC3 and then directed toward a lysosome. Fusion creates the environment in which the enclosed material can be broken down. The resulting components can be returned to the cytosol and reused. This process is not limited to routine housekeeping. Nutrient depletion can activate autophagy as a way to preserve viability. Intracellular infections can engage it as part of a protective response. Autophagy also participates in intracellular repair, and under some conditions, it can contribute to programmed cell death. The important distinction is the direction of the cargo. In heterophagy, the lysosome receives material that entered from outside. In autophagy, the cell packages material from within and sends it to the same degradative compartment. The endpoint is shared, but the route and the biological question are different. We have now seen 3 ways large cargo reaches the lysosome: endocytosis, phagocytosis, and autophagy. A different disposal machine handles many soluble cytosolic proteins.
The proteasome handles a different class of cargo. It is a cytosolic disposal complex for denatured, misfolded, or otherwise tagged proteins, including regulatory proteins whose lifespan must be controlled. Many proteins destined for destruction are marked by covalent attachment of a small protein called ubiquitin. The tagging process uses 3 functional steps. E1, E2, and E3 ubiquitin ligases help identify the substrate and build a polyubiquitin signal. The marked protein is then unfolded and fed into the proteasome, a multi-subunit cylinder with protease activities facing a hollow central chamber. Inside that chamber, the protein is cut into short peptide fragments, roughly 6 to 12 amino acids long. Those fragments can be broken down further, and their amino acids can be recycled. The proteasome is therefore selective. It does not receive every large particle that enters the cell, and it does not digest an entire worn-out organelle. It recognizes a protein-level disposal signal. This also gives the cell a way to regulate biology. Removing a signaling molecule or transcription factor can switch a pathway off. In antigen-presenting cells, proteasomal peptides can enter the major histocompatibility class 1 or class 2 pathways and help drive adaptive immunity. The lysosome and the proteasome are both disposal systems, but their cargo rules are not interchangeable. It is useful to place the disposal routes side by side. Lysosomes are membrane-bound acidic compartments. They receive material from endosomes and phagosomes, and they receive autophagosomes containing worn-out organelles or large protein complexes. Their enzymes digest a broad range of macromolecules. Proteasomes remain in the cytosol and focus on selected proteins. A ubiquitin tag identifies the substrate. The protein is unfolded, and the proteasome cuts it into short peptides. The route is controlled by molecular recognition rather than by enclosing a large piece of cargo. Peroxisomes are another specialized compartment. They contain oxidative enzymes, including catalase and peroxidase, and have an important role in breaking down very long-chain fatty acids, generating hydrogen peroxide as part of the process. These systems should not be collapsed into one generic idea of cellular garbage disposal. The cell asks different questions. Is this material arriving from outside? Is it a worn-out organelle? Is it one misfolded cytosolic protein whose lifetime needs to end? Or is it a hydrophobic fatty acid requiring oxidative processing? The answer
determines the compartment. This separation protects the cell while allowing molecular components to be recovered. The final step is to see how manufacture and disposal form one continuous cycle.
Now put the whole pathway together. A protein intended for the cytosol can be made on a free ribosome and later removed through the ubiquitin proteasome system. A protein intended for a membrane or for secretion enters the rough ER, folds and is inspected, then travels to the Golgi for further modification and sorting. From the trans-Golgi network, cargo can reach the plasma membrane, a secretory vesicle, another organelle, or the lysosome. Material entering from outside can pass through endosomes. A microbe can become a phagosome and then a phagolysosome. A worn-out organelle can enter an autophagosome and meet the same lysosomal machinery. The cell is therefore not just producing structures and leaving them in place. It is continually checking them, moving them, using them, and removing them when their lifetime ends. ER stress shows what happens when production and quality control fall out of balance. Lysosomal and proteasomal pathways show how the cell restores usable building blocks while limiting harmful accumulation. That is the logic of cellular housekeeping: compartmentalized work, controlled routing, and continuous renewal. We can now ask what supplies the energy and metabolic intermediates that keep this entire logistics system running. That takes us next to cellular metabolism and mitochondrial function.